So here's something worth thinking about as you settle in tonight. Right now, at this very moment, without any conscious effort on your part, your body is doing something almost impossibly complex. Your heart is beating, and you didn't tell it to. Your lungs are drawing in air, and you're not thinking about it. Your eyes, if they're still open, are taking in light and converting it into electrical signals that travel along delicate cables of living tissue into the back of your skull, where somehow, through a process that is still not fully understood, those signals become the experience of seeing. And if you shift your weight right now, even slightly, dozens of muscles will fire in precisely the right sequence, coordinated by a network of communication so fast and so intricate that the world's most advanced supercomputers can't fully simulate even a small portion of it. That network is your nervous system. And tonight, we're going to talk about how it works, how we figured out how it works, and why the story of that figuring out is one of the most fascinating, frustrating, and genuinely astonishing journeys in the history of science. We'll take it slow. There's no rush at all. If you drift off somewhere along the way, that's perfectly fine. Your nervous system will keep running beautifully without you paying any attention to it, which is, when you think about it, one of the most remarkable things about it. Let's start where most big scientific stories start, which is with people getting it completely wrong. Because for most of human history, nobody had any idea that the brain was the organ responsible for thinking, feeling, and experiencing the world. That might sound strange to us now, but think about it from the perspective of someone living 3,000 or 4,000 years ago. If you put your hand on your chest, you can feel your heart beating. It speeds up when you're scared. It races when you're in love. It seems to respond to your emotions in a way that's immediate and obvious. The brain, on the other hand, just sits up there in your skull, soft and squishy and quiet. You can't feel it doing anything. It doesn't pulse. It doesn't react in any way you can sense from the outside. So it's actually pretty reasonable that many ancient civilizations concluded the heart was where the important stuff happened. The ancient Egyptians are a perfect example. They were incredibly sophisticated in many ways. Their engineering, their medicine, their understanding of anatomy from the embalming process. They'd been opening up bodies for thousands of years. And yet, when they mummified someone for the afterlife, they carefully preserved the heart, the lungs, the liver, the intestines, but the brain? They pulled it out through the nose with a hook and threw it away, just discarded it. They didn't think it was important. The heart was the seat of the soul, the organ of thought and feeling and identity. The brain was just filler, packing material inside the skull. Now, to be fair, not everyone got it wrong. There were dissenters. Alcmaeon of Croton, a Greek physician and philosopher who lived around 500 BCE, is one of the first people we know of who argued that the brain was the organ of sensation and thought. He apparently dissected animals and traced the optic nerves from the eyes back to the brain, and he concluded that the brain was where perception happened. Hippocrates, the father of medicine, agreed. He wrote explicitly that the brain is the organ that allows us to think, see, hear, and distinguish between what is pleasant and what is ugly. That's a remarkably clear statement for the 5th century BCE. But here's the thing. They lost the argument, at least for a while, because then along came Aristotle, and Aristotle was arguably the most influential thinker in the Western world for nearly 2,000 years. And Aristotle was a heart guy. He looked at the brain and noted that it was bloodless, that it was cold to the touch, and that poking it didn't seem to produce any sensation. The heart, by contrast, was warm. It was active. It was obviously central to life. Aristotle concluded that the heart was the seat of intelligence and sensation, and the brain was basically a cooling system, a radiator for the blood. He thought the brain existed to cool the heat generated by the heart, and because Aristotle's authority was so enormous, this view dominated for centuries. It's actually a beautiful example of how a brilliant person being wrong about something can set back understanding for an incredibly long time. Aristotle was right about an astonishing number of things, but on this one, he was completely, spectacularly wrong, and his wrongness cast a very long shadow. The person who started to pull things back in the right direction was a physician named Galen, Claudius Galenus, born around 109 CE in Pergamon, in what is now Turkey. Galen is one of the most important figures in the history of medicine, and he lived one of the most interesting lives of any scientist in antiquity. He started his medical career treating gladiators, actual gladiators, the men who fought and bled and died in the arenas of the Roman world. And this was, in a grim way, an incredible education, because gladiator wounds gave Galen a window into the living human body that most physicians never got. He could see what happened when nerves were severed. He could observe which injuries produced paralysis and which didn't. He watched men lose the ability to move an arm or a leg or to speak, and he could correlate those losses with the specific location of the wound. Galen also performed extensive dissections and experiments on animals, pigs, goats, and especially Barbary macaques, a type of monkey. And through this work, he demonstrated something crucial. He showed that the brain, not the heart, controlled movement and sensation. He proved this with a series of experiments that were, honestly, pretty brutal by modern standards, but scientifically groundbreaking. He would expose the brains and spinal cords of living animals and systematically cut or compress different parts, observing which functions were lost. He showed that severing nerves in certain locations produced specific paralyses. He demonstrated that damage to the brain could abolish sensation, movement, even consciousness, while the heart kept beating. This was huge. Galen essentially proved, through experimental evidence, that the nervous system was the body's command and control network. He traced nerves from the brain down through the spinal cord and out to the muscles and sense organs. He distinguished between sensory nerves, the ones that carry information from the body to the brain, and motor nerves, the ones that carry commands from the brain to the muscles. That distinction, sensory versus motor, is one of the most fundamental concepts in neuroscience, and Galen identified it nearly 2,000 years ago. But here's where it gets complicated, because Galen also got some important things wrong, and his mistakes, like Aristotle's, persisted for centuries. Galen believed that nerves were hollow tubes, and that they worked by carrying a fluid. He called it animal spirits, from the brain to the body and back. In his model, the brain manufactured this fluid, this pneuma, in the ventricles, the fluid-filled chambers inside the brain, and pumped it through the nerves the way the heart pumps blood through the arteries. Sensation happened when animal spirits flowed from the sense organs up to the brain. Movement happened when animal spirits flowed from the brain down to the muscles. Now, this is wrong. Nerves aren't hollow, and they don't carry fluid in any functionally meaningful way. But it's a brilliantly logical guess given what Galen could observe. The ventricles of the brain really do exist, and they really are filled with fluid, cerebrospinal fluid. Nerves really do look like they could be tiny tubes if you squint, and the model actually explained a lot of the observations. If you cut a nerve, the function downstream is lost. Just as if you cut a pipe, the water stops flowing beyond the cut. It was elegant, it was consistent with the evidence, and it was wrong. And this model, the hollow nerve, animal spirits model, dominated Western medical thinking for well over 1,000 years. Through the Middle Ages, through the Renaissance, when anatomists like Andreas Vesalius in the 1540s corrected many of Galen's anatomical errors, but still largely accepted the basic framework of animal spirits flowing through nerves. It wasn't until the 17th and 18th centuries that people started seriously questioning the whole paradigm, and what finally overturned it was one of the most dramatic and contentious scientific discoveries of the age. It involved dead frogs, an argument between two Italian scientists that got very personal, and the entirely accidental discovery that would eventually give us the electric battery. And it started in a laboratory in Bologna in the 1780s, with a physician named Luigi Galvani and a plate of frog legs. Galvani was a professor of anatomy at the University of Bologna, and the story of how he made his great discovery is one of those beautiful accidents that science is full of. The details have been told and retold so many times that separating the pure facts from the legend is a little tricky, but here's the gist. Galvani had been studying frogs, dissected frog preparations specifically, and at some point, he or one of his assistants noticed something strange. When a metal instrument touched the nerve of a dissected frog leg, the leg twitched. It kicked, even though the frog was dead, even though it had been cut apart and was lying on a table. Touch the nerve with metal, and the muscles contracted as if the frog were alive. Now, this wasn't the first time anyone had noticed something like this. People had been playing around with static electricity and its effects on muscles for decades by this point. But Galvani was systematic about it. He experimented extensively throughout the 1780s, trying different metals, different conditions, different preparations. And eventually, he came to a bold conclusion. The frog's body itself, he argued, contained electricity. The nerves and muscles were producing their own electrical charge. He called it animal electricity, and he believed he had discovered the force that Galen's animal spirits were trying to describe. The vital force that animated living things wasn't some mysterious fluid. It was electricity. Electricity generated by the living tissue itself. This was revolutionary. And it was also, in certain important respects, correct. But it immediately sparked one of the most famous scientific feuds of the 18th century, because another Italian scientist, Alessandro Volta, a physicist at the University of Pavia, disagreed sharply with Galvani's interpretation. Volta's argument was clever and, at first glance, devastating. He pointed out that in most of Galvani's experiments, the frog leg twitched when two different metals were involved. Say, a copper hook holding the frog and an iron railing it was touching. Volta argued that the electricity wasn't coming from the frog at all. It was being generated by the contact between the two different metals, and the frog's leg was just acting as a detector, a sensitive indicator of the current flowing between them. The frog wasn't producing electricity. It was responding to electricity produced by the metals. And to prove his point, Volta built something extraordinary. In 1800, he stacked alternating discs of zinc and copper, separated by cardboard soaked in brine, and produced the first device in history that could generate a steady, continuous flow of electric current. He called it the pile. We call it a battery, the voltaic pile. It was the first battery ever made, and it came directly out of this argument about frog legs. The unit of electrical potential, the volt, is named after Volta, and for a while, it seemed like Volta had won the argument. The electricity was coming from the metals, not the animal. But here's the thing, Galvani was also right. Both of them were right about different aspects of the problem, and history has vindicated them both. Volta was correct that contact between dissimilar metals can generate electricity. That's how batteries work to this day. But Galvani was also correct that living tissue generates its own electrical signals. Nerves really do produce electricity. Muscles really do produce electricity. The frog leg wasn't just a passive detector. It was an electrically active tissue responding to electrical stimulation. Galvani had glimpsed something genuine and profound, that the language of the nervous system is electrical, but the tools to prove it definitively wouldn't exist for another half-century. And the resolution of this debate would require a completely new way of thinking about what a nerve actually is. And that brings us to one of the most important tools in the entire history of biology, the microscope. Because as long as people were looking at nerves with the naked eye, there was only so much they could figure out. A nerve, to the unaided eye, looks like a thin, whitish cord. You can trace it from one place to another. You can cut it and observe that function is lost. But you can't see its internal structure. You can't see what it's made of. And until you can do that, you're essentially trying to understand how a computer works by looking at the outside of the case. The microscope changed everything. And the story of what people saw when they finally looked at nervous tissue under magnification is one of the most contentious, personality-driven, and ultimately beautiful stories in all of science. It's a story that revolves around two men who despised each other, who fundamentally disagreed about what they were looking at through their microscopes, and who shared a Nobel Prize in 1906 under circumstances so awkward that the ceremony itself became legendary. Their names were Camillo Golgi and Santiago Ramon y Cajal, and their rivalry shaped the entire field of neuroscience. Golgi was Italian, born in 1843 in a small town in Lombardy. He trained as a physician and became fascinated by histology, the study of tissues under the microscope. And in the early 1870s, while working in a hospital kitchen that he'd converted into a makeshift laboratory, he couldn't afford a proper lab, Golgi developed a staining technique that was nothing short of miraculous. The problem with looking at brain tissue under a microscope is that it's basically transparent. Everything is tangled together in a dense, soupy mess, and you can't distinguish one cell from another. It's like trying to trace a single strand of spaghetti in a bowl of cooked pasta. You just can't see where one ends and another begins. Golgi's technique, which he called the black reaction, solved this in the most elegant way imaginable. He soaked brain tissue in potassium dichromate and then in silver nitrate, and something remarkable happened. The silver compound would impregnate only a small, seemingly random fraction of the neurons, maybe 1 or 2%, and stain them completely, in vivid black, against a clear yellow background. So instead of seeing the entire hopeless tangle, you could suddenly see individual cells in their entirety, with all their branches and extensions, standing out as clearly as trees silhouetted against a winter sky. It was like someone had switched on a light in a room that had been dark for centuries. And what Golgi saw was extraordinary. Neurons, nerve cells, weren't simple blobs. They had complex, elaborate structures. From the cell body, long, thin projections extended outward, branching and rebranching, like the limbs of a tree. Some of these branches were short and bushy. We now call these dendrites. And typically one projection was much longer than the others, stretching far away from the cell body, sometimes for enormous distances. This is the axon, the main output cable of the neuron. But Golgi interpreted what he was seeing in a particular way. He looked at those tangled branches and concluded that the entire nervous system was a continuous network, a single, unbroken web of connected tissue, like a fishing net, where all the threads are fused together. He called this the reticular theory. In Golgi's view, the nervous system wasn't made up of individual cells. It was one giant, interconnected meshwork. And then, along came Cajal. Santiago Ramón y Cajal was Spanish, born in 1852, in a small town in Aragon. His father was a physician who wanted Santiago to follow in his footsteps, but the young Cajal was, by his own account, a terrible student and a bit of a troublemaker. He wanted to be an artist. He was obsessed with drawing, with capturing the shapes and forms of the natural world. His father eventually persuaded him into medical school, but that artistic eye, that ability to see and reproduce intricate visual detail, turned out to be the secret weapon that would make Cajal arguably the most important neuroscientist who ever lived. Cajal learned about Golgi's staining technique in the late 1880s, and when he applied it to brain tissue, he was transfixed. He spent the next several decades bent over a microscope, hour after hour, day after day, drawing what he saw with extraordinary precision and beauty. His drawings of neurons are still used in textbooks today, and they're not just scientifically accurate, they're genuinely gorgeous, works of art. But here's where the disagreement comes in. Cajal looked at the same stained neurons that Golgi had looked at, and he reached the opposite conclusion. Where Golgi saw a continuous network, Cajal saw individual cells. He argued that neurons were discrete, separate units, that they came very close to each other, sometimes almost touching, but they didn't actually fuse. There were tiny gaps between them. Each neuron was its own independent entity, receiving signals through its dendrites, processing them in its cell body, and sending signals out along its axon to the next neuron. But the connection between them was not a physical fusion. It was a gap, a junction, across which some kind of signal had to pass. This was called the neuron doctrine, and it was a radically different way of thinking about the nervous system. Instead of one continuous network, the brain was a vast collection of individual cells, each one a tiny processing unit, communicating with its neighbors across tiny spaces. The argument between Golgi and Cajal was fierce, personal, and lasted for years. And in 1906, in what might be the most awkward Nobel Prize ceremony in history, they were both awarded the Nobel Prize in Physiology or Medicine. Together, they had to share it. And at the ceremony in Stockholm, each man used his Nobel lecture to argue that the other one was wrong. Golgi went first, and he spent much of his time defending the reticular theory and attacking the neuron doctrine. Then Cajal got up and systematically demolished Golgi's position. It was, by all accounts, deeply uncomfortable for everyone in the room, and history has been unambiguous about the verdict. Cajal was right. The nervous system is made up of individual cells, neurons, that communicate across tiny gaps. Those gaps would eventually be named synapses, a term coined by the English physiologist Charles Sherrington in 1897, from the Greek word meaning to clasp together. And the discovery and understanding of what happens at those synapses, how one neuron talks to the next, would become one of the central dramas of 20th century neuroscience. But before we get to the synapse, we need to understand the signal itself, the electrical impulse that travels along a neuron. Because remember that Galvani had shown back in the 1780s that nerves are electrical. But knowing that nerves use electricity and understanding exactly how they use it are two very different things. And figuring out the mechanism, the actual nuts and bolts physics and chemistry of how a nerve fires, took another 150 years, and it required some genuinely brilliant experiments involving a very unusual animal. Let's talk about the squid, specifically the squid giant axon, and I mean giant literally. Most axons in the human body are microscopically thin, a few thousandths of a millimeter in diameter. You'd need a powerful microscope just to see them. But certain species of squid have axons that are enormous, up to 1 millimeter in diameter, about the width of the lead in a mechanical pencil. You can actually see them with the naked eye. And the reason squid have these monster axons is speed. The squid uses them to control its jet propulsion escape response, that explosive backward jet of water it uses to flee from predators. The signal to fire that escape jet has to travel very fast, and in the simple nervous system of a squid, the way to make a signal travel faster is to make the axon thicker. A thicker cable means less electrical resistance, which means the signal propagates more quickly. It's the same basic principle as using a thicker copper wire to carry more current. Now, for scientists trying to understand how nerve signals work, the squid giant axon was an absolute gift. Because it's big enough to stick electrodes into, you can impale the thing with tiny glass tubes filled with conducting solution and directly measure the electrical voltage inside the axon while it's firing. Try doing that with a human nerve fiber a few thousandths of a millimeter across. In the 1930s and 40s, it was basically impossible. But with a squid axon, you could do it. And the people who did it, who cracked the code of how nerve impulses actually work, were two British physiologists named Alan Hodgkin and Andrew Huxley. Their work, carried out primarily at the Marine Biological Association Laboratory in Plymouth, England, in the late 1930s, and then again after the war in the late 1940s, is one of the towering achievements of 20th century biology. They won the Nobel Prize for it in 1963. And honestly, the work is so elegant, so cleverly designed, and so beautifully analyzed that it's worth spending some real time on. Here's what they were working with. You've got a nerve cell, a neuron, and it has a membrane, a thin fatty layer that separates the inside of the cell from the outside. And across that membrane, there's an electrical voltage. The inside of the neuron, when it's resting, when it's not doing anything, is slightly negatively charged compared to the outside. The difference is about 70 millivolts, roughly a 20th of a volt. That doesn't sound like much, but the membrane is incredibly thin, only about 5 nanometers thick, so the electric field across it is actually enormous, something like 14 million volts per meter if you scaled it up. That's a staggeringly intense electric field packed into a vanishingly thin space. Now, where does this voltage come from? This is where the chemistry gets beautiful. The fluid inside a neuron has a different chemical composition from the fluid outside it. Inside, there's a lot of potassium ions and not much sodium. Outside, it's the reverse. Lots of sodium, not much potassium. These differences are maintained by tiny molecular machines embedded in the membrane called sodium-potassium pumps. Each pump grabs 3 sodium ions from inside the cell, shoves them out, and simultaneously grabs 2 potassium ions from outside and pulls them in. It does this over and over, thousands of times per second, using energy from ATP, the cell's universal energy currency. So the pump creates an imbalance. Sodium concentrated outside, potassium concentrated inside. But here's the clever part. The membrane also has channels, tiny protein pores that can open and close. When the cell is resting, some potassium channels are open, but most sodium channels are closed. And potassium, because it's concentrated inside, naturally wants to leak out through those open channels. As positively charged potassium ions leak out, they leave behind negatively charged proteins and other molecules that can't fit through the channels. This creates a charge separation. Negative inside, positive outside. That's the resting potential. That negative 70 millivolts. So far, so good. The cell is sitting there, negatively charged on the inside, ready and waiting. And now something happens. Maybe a signal arrives from another neuron. Maybe a sensory receptor is stimulated. Whatever it is, it causes the membrane voltage at one point on the axon to become slightly less negative. It rises from -70 millivolts towards zero. And if it rises enough, if it crosses a critical threshold, about -55 millivolts, something dramatic happens. The sodium channels at that point suddenly snap open, and when they open, sodium comes flooding in. Remember, sodium is concentrated outside, and the inside is negatively charged, so sodium is being pulled in by both the concentration difference and the electrical gradient. It rushes in through the newly opened channels, and the voltage at that point doesn't just reach zero, it actually overshoots, going all the way to about +40 millivolts. The inside of the cell at that spot has gone from negative to positive in about 1 millisecond. That's the action potential. That's the nerve impulse, that rapid, dramatic reversal of voltage. But it doesn't stop there. Almost immediately, within a millisecond or so, the sodium channels slam shut again, and a different set of channels, the potassium channels, open wider. Now potassium rushes out, driven by the concentration gradient and by the fact that the inside has just become positively charged. This outflow of positive potassium ions drives the voltage back down, actually overshooting slightly in the negative direction, before the whole thing settles back to the resting potential. The sodium-potassium pumps then clean up, restoring the original ion distribution. The whole event, the spike, the action potential, lasts about 2 milliseconds. And here's what's really beautiful about this. That spike of voltage at one point on the axon doesn't just sit there. It triggers the same process in the neighboring stretch of membrane. The rush of sodium ions into one region creates a small current that depolarizes the adjacent region just enough to trigger its sodium channels to open, and that triggers the next region, and the next. The action potential propagates down the axon like a wave, like a line of dominoes falling, where each domino knocks over the next. And because each domino is powered by its own local reserves of sodium and potassium, the signal doesn't get weaker as it travels. It's regenerated at every point along the way. An action potential that travels a meter, from your spine to your fingertips, say, arrives with exactly the same strength as when it started. That's a fundamentally different mechanism from a simple electrical current flowing through a wire, where the signal degrades with distance. The nerve does something more clever. It actively reconstructs the signal at every point. Hodgkin and Huxley worked all of this out. They measured the currents flowing through squid axon membranes with astonishing precision. They figured out the separate contributions of sodium and potassium. They wrote down mathematical equations describing how the channels open and close as a function of voltage and time. And then they took those equations and, using a hand-cranked mechanical calculator, this was before computers were widely available, they computed what the action potential should look like, based purely on their model of the ion channels. It took them weeks of grinding calculation, and the predicted action potential matched the measured one almost perfectly. The shape, the timing, the speed of propagation, it all matched. That's the gold standard in science. Build a model, make a prediction, check it against reality. Hodgkin and Huxley nailed it. Now, here's something I glossed over that's worth coming back to, because it solves a problem you might have been thinking about. If the action potential propagates along the axon by triggering each adjacent region in sequence, the speed of propagation depends on how quickly the current can spread from one region to the next, which depends on the diameter of the axon. Thicker axons conduct faster, which is why squid have those giant axons for their escape reflex. But you can't just make every axon thick. Your brain has something like 86 billion neurons. and many of them need long axons to connect distant parts of the brain or to reach down the spinal cord to your toes. If all those axons were as thick as squid axons, your head would need to be the size of a swimming pool. So vertebrates, animals with backbones, including you, evolved a different solution. It's called myelination, and it's one of the most elegant engineering solutions in all of biology. Instead of making the axon thicker, your nervous system wraps the axon in a fatty insulating sheath called myelin. This is produced by specialized cells, oligodendrocytes in the brain and spinal cord, Schwann cells in the peripheral nerves. These cells literally wrap themselves around the axon, layer upon layer, like electrical tape around a wire. And this insulation prevents the ion channels from doing their thing along most of the axon's length. The action potential can't regenerate under the myelin. But here's the trick. The myelin isn't continuous. It has regularly spaced gaps, like the links of a sausage chain. These gaps are called nodes of Ranvier, named after the French anatomist Louis-Antoine Ranvier, who described them in the 1870s. At the nodes, the axon membrane is exposed and densely packed with sodium channels. So the action potential jumps from node to node, skipping over the insulated segments in between. This is called saltatory conduction, from the Latin word saltare, meaning to jump, and it's dramatically faster than continuous conduction. A myelinated axon can conduct signals at up to 120 meters per second. That's over 260 miles per hour, while being much thinner than an unmyelinated axon conducting at the same speed would need to be. Your myelinated motor neurons can send a signal from your spinal cord to your foot in about 1/100th of a second. And this is why demyelinating diseases are so devastating. Multiple sclerosis, for example, is a condition where the immune system mistakenly attacks the myelin sheath, stripping the insulation from neurons in the brain and spinal cord. When the myelin is damaged, the action potential can no longer jump efficiently from node to node. Signals slow down, become erratic, or fail altogether. Depending on which neurons are affected, the symptoms can include numbness, weakness, vision problems, difficulty with coordination, fatigue, and a whole constellation of other issues. The wiring is still there, but the insulation is damaged, and that makes all the difference. All right, so now we know how a signal travels along a single neuron, the action potential, this beautiful cascade of sodium and potassium channels opening and closing in sequence, regenerating the signal at every point, jumping from node to node along myelinated fibers. But a single neuron by itself doesn't do much. The real magic of the nervous system happens at the connections between neurons, at those tiny gaps that Cajal predicted and Sherrington named, the synapses. And the story of how we figured out what happens at the synapse is one of the best stories in all of neuroscience, because it involves a dream. Literally, a dream. And it starts with a question that might seem simple, but turned out to be deeply controversial for decades. When an electrical signal reaches the end of one neuron, how does it cross the gap to the next neuron? Is the signal purely electrical? Does the current just jump across like a spark? Or is there a chemical involved? Does the first neuron release some substance that triggers the second neuron to fire? This was a genuine scientific controversy in the early 20th century. On one side were the sparks, the people who thought transmission across the synapse was electrical. On the other side were the soups, the people who thought it was chemical. The sparks had some logic on their side. Electrical transmission would be fast. The signal in the nerve was electrical, so why wouldn't the transmission at the junction also be electrical? The soups had a different kind of logic. They pointed out that certain drugs and chemicals clearly affected nerve transmission, things like curare, which paralyzes muscles, or adrenaline, which speeds the heart, and these effects were hard to explain if the synapse was purely electrical. The person who settled the argument, at least for the most important class of synapses, was an Austrian physiologist named Otto Loewi, and the way he did it is one of the most famous stories in the history of biology. Loewi had been thinking about the chemical versus electrical question for years. The idea for the definitive experiment had been floating around in his mind, half-formed, never quite crystallizing. And then, in 1921, he had a dream. He woke up in the middle of the night with the experimental design clear in his mind. He scribbled some notes on a scrap of paper and went back to sleep. The next morning, he looked at his notes and couldn't read them. He couldn't remember the dream. He was devastated. But the next night, the dream came back. This time, Loewi didn't bother with notes. He got out of bed, went straight to his laboratory at 3 a.m. and performed the experiment. And it worked. Here's what he did. He took 2 frog hearts and kept them alive in a bath of saline solution. He stimulated the vagus nerve of the first heart, which slowed it down. The vagus nerve is the nerve that tells the heart to beat more slowly, and we'll talk much more about it later. Then he took the saline solution that the first heart had been sitting in, the fluid that had bathed that heart while its vagus nerve was being stimulated, and he transferred it to the second heart, and the second heart slowed down too, even though nobody had touched its vagus nerve, even though there was no electrical connection between the 2 hearts. Think about what that means. Something had been released by the first heart's vagus nerve into the fluid, a chemical substance, and that substance, when it reached the second heart, produced the same effect. The nerve wasn't communicating through electricity at the synapse. It was releasing a chemical messenger. Loewi called this substance Vagusstoff, vagus substance. We now know it as acetylcholine, and it was the first neurotransmitter ever identified. This experiment, elegant in its simplicity, cracked open one of the biggest questions in neuroscience. And it won Loewi the Nobel Prize in 1936, shared with Henry Dale, who had done crucial work identifying acetylcholine and demonstrating that it was released by motor neurons at the junction with muscles, the neuromuscular junction. Now, it's worth noting that the sparks weren't entirely wrong. Some synapses in the nervous system really are electrical. They're called gap junctions, and they consist of tiny channels that directly connect the insides of adjacent neurons, allowing electrical current to flow straight through. These are very fast, and they're found in circuits where speed is absolutely critical, like certain escape reflexes. But the vast majority of synapses in your brain, and there are somewhere between 100 trillion and 1 quadrillion of them, are chemical. They work by releasing neurotransmitters. So here's how a chemical synapse works, step by step, and it's worth walking through, because it's one of those mechanisms that's incredibly clever once you see it all laid out. An action potential comes racing down the axon and arrives at the very tip, the axon terminal, sometimes called the synaptic bouton. And at the tip, the arrival of the action potential triggers a specific set of calcium channels to open. Calcium ions, which are concentrated outside the cell, rush in. And this influx of calcium triggers a cascade of events inside the terminal. Tiny bubbles, vesicles, that are filled with neurotransmitter molecules are sitting there in the terminal, preloaded and ready to go. The calcium signal causes these vesicles to move to the edge of the membrane and fuse with it, releasing their contents into the synaptic cleft, that tiny gap between the 2 neurons. The gap is only about 20 nanometers wide, smaller than a virus. The neurotransmitter molecules diffuse across this tiny gap, which takes only microseconds. It's such a short distance. And they bind to receptor proteins on the surface of the next neuron. These receptors are like locks, waiting for the right key. When the neurotransmitter binds, the receptor changes shape and opens an ion channel, allowing ions to flow into or out of the second neuron. And depending on which ions flow and in which direction, this either pushes the second neuron closer to firing its own action potential, an excitatory effect, or pulls it further away from firing, an inhibitory effect. And here's the thing that makes this system so powerful. A single neuron in your brain receives input from thousands of other neurons, sometimes tens of thousands. Some of those inputs are excitatory, nudging it toward firing. Others are inhibitory, pulling it back. The neuron integrates all of these inputs. It essentially adds up all the excitation and subtracts all the inhibition. And if the net result crosses that threshold, it fires its own action potential. If not, it stays quiet. This integration of thousands of competing inputs, happening at every one of the 86 billion neurons in your brain, simultaneously, continuously, that's computation. That's how your brain thinks. Not through any single dramatic event, but through this vast, rolling chorus of tiny chemical whispers, billions of synapses chattering away, inhibiting and exciting each other, constantly adjusting the network's activity. And the neurotransmitters themselves, we've discovered dozens of them by now, and they play fascinatingly different roles. Acetylcholine, the one Lowy discovered, is the main neurotransmitter at the neuromuscular junction. It's what makes your muscles contract. It's also important in the brain for attention and memory. Dopamine, which you've probably heard of, is involved in reward, motivation, and movement. Parkinson's disease is caused by the loss of dopamine-producing neurons in a specific part of the brain called the substantia nigra, and the movement difficulties that result are a direct consequence of losing this chemical signal. Serotonin influences mood, sleep, and appetite. Many antidepressant medications work by increasing the amount of serotonin available at synapses. Norepinephrine is involved in alertness and the stress response. GABA, gamma-aminobutyric acid, is the main inhibitory neurotransmitter in the brain, the one that calms things down and keeps neural activity from spiraling out of control. Glutamate is the main excitatory neurotransmitter, the one that gets things going. And what's really fascinating is that the balance between excitation and inhibition is absolutely critical. Too much excitation and you get seizures, neurons firing uncontrollably in cascading waves of activity. Epilepsy, in many forms, involves disruptions to this balance. Too much inhibition and activity grinds to a halt. The brain is constantly walking a tightrope between these two extremes, and the fact that it almost always gets it right is a testament to how exquisitely tuned the system is. Now, there's another layer of complexity here that's worth mentioning, because it changes the picture in a really important way. When I said that neurotransmitters bind to receptors and open ion channels, that's true for one class of receptors, the fast-acting ones, called ionotropic receptors. The neurotransmitter binds, the channel opens, ions flow, and the effect happens in milliseconds. But there's another class of receptors, metabotropic receptors, that work completely differently. These receptors don't open ion channels directly. Instead, when a neurotransmitter binds to them, they trigger a cascade of chemical signals inside the receiving neuron. These internal signals can modify the cell's behavior in ways that last much longer, seconds, minutes, even permanently. They can change which genes are being expressed, alter the sensitivity of other receptors, strengthen or weaken synaptic connections, and fundamentally reshape how the neuron responds to future inputs. And this is where we start to touch on something that has dominated neuroscience for the last several decades: the idea that synaptic connections are not fixed, They change. They strengthen when they're used frequently and in meaningful patterns. They weaken when they're not. The brain is not a hardwired circuit board. It's a living, changing, constantly adapting network, and the changes at the synaptic level, the strengthening and weakening of connections between neurons, are believed to be the fundamental mechanism underlying learning and memory. But that's a story that deserves its own space, and we'll come back to it. For now, let's shift our attention to something you're experiencing right at this moment, probably without thinking about it much: your senses. Because everything we've been talking about, neurons, action potentials, synapses, neurotransmitters, all of this machinery exists, ultimately, to do two things: to take in information from the world and to act on it. And the taking in part, the sensory side, is one of the most elegant pieces of engineering in the entire body. Start with something simple: touch. Right now, you can probably feel the weight of a blanket on your skin, or the texture of a pillow against your cheek, or the pressure of your body against the mattress. That sensation, something so mundane you barely notice it, requires a chain of events that, when you lay it out, is genuinely remarkable. In your skin, there are specialized receptor cells, millions of them, scattered throughout your body at different densities. Your fingertips have an extraordinarily high concentration. The skin on your back has far fewer. That's why you can distinguish between two pinpricks a couple of millimeters apart on your fingertip, but not on your back, where they'd need to be centimeters apart before you could tell they were two separate points. These receptors come in different types, each sensitive to a different kind of stimulus. Merkel cells respond to sustained pressure. They're what let you feel the shape of an object you're holding. Meissner's corpuscles respond to light, fluttery touch, and low-frequency vibrations. They're concentrated in your fingertips and lips, and are responsible for your ability to read Braille or feel the texture of fabric. Pacinian corpuscles, which are deeper in the skin, respond to rapid vibrations and deep pressure. Ruffini endings respond to sustained pressure and stretching of the skin. And there are free nerve endings throughout your skin that respond to temperature and pain. Each of these receptors, when stimulated, converts its particular type of physical stimulus into an electrical signal, an action potential, that travels along a sensory nerve fiber toward the spinal cord and up to the brain. This conversion process is called transduction, and it's a beautiful example of biological engineering. Take a Pacinian corpuscle, for instance. It's built like a tiny onion, concentric layers of membrane surrounding a nerve ending at the center. When pressure deforms the corpuscle, it stretches the membrane of the nerve ending, which opens mechanically gated ion channels. Ions flow through, the membrane depolarizes, and if the depolarization is large enough, it triggers an action potential. Physical force has been translated into electrical signal. The language of touch has been converted into the language of the nervous system. And here's something clever about how the intensity of a stimulus is encoded. Remember, an action potential is an all-or-nothing event. It either fires or it doesn't. And when it fires, it's always the same size. So how does your brain know the difference between a gentle caress and a firm handshake? Not by the size of the individual action potentials, but by their frequency. A stronger stimulus causes the receptor to fire more action potentials per second. A light touch might generate 10 action potentials per second. A firm press might generate 100. And some stimuli are so intense that the receptor fires at 500 or more per second. Your brain reads the rate of firing and interprets it as intensity. It's frequency modulation, like an FM radio signal. The message is in the rate, not the amplitude. Now, let's talk about something more complex. Let's talk about vision, because the eye is one of the most extraordinary sensory organs in all of biology, and the way it interfaces with the brain is breathtaking. Light enters your eye through the cornea, passes through the pupil, which adjusts its size to control how much light gets in, and is focused by the lens onto the retina, a thin layer of tissue at the back of the eye. And here's where things get really interesting, because the retina is not just a simple screen. It's actually a piece of your brain. Developmentally, the retina grows out from the brain during embryonic development. It's neural tissue, packed with neurons, and it does a significant amount of processing before it ever sends a signal to the rest of the brain. The light-detecting cells in the retina are called photoreceptors, and there are two main types, rods and cones. You have about 120 million rods and about 6 million cones. Rods are incredibly sensitive. They can detect a single photon of light, but they don't distinguish colors. They're your night vision system, the ones doing the work when you're lying in a dimly lit room. Cones are less sensitive, but they come in 3 varieties, each sensitive to a different range of wavelengths, roughly red, green, and blue. Your perception of color comes from your brain comparing the relative activation of these 3 cone types. Every color you've ever seen, every sunset and painting and autumn leaf, is your brain's interpretation of different ratios of activation in 3 types of cone cells. That's it, 3 channels. And from those 3 channels, you perceive millions of distinct colors. The brain is doing something extraordinary with very limited raw data. Now, here's where the engineering gets really clever. The photoreceptors don't send their signals directly to the brain. They connect to a layer of intermediate neurons, bipolar cells, which connect to another layer called ganglion cells. And it's the ganglion cells whose axons bundle together to form the optic nerve, which carries visual information from the eye to the brain. But those intermediate layers aren't just passing signals along passively. There are lateral connections, horizontal cells and amacrine cells, that connect photoreceptors and bipolar cells to their neighbors. And these lateral connections perform computation. They're comparing the activity of nearby cells, enhancing contrasts, detecting edges, responding to motion. The retina is doing sophisticated image processing before the signal even leaves the eye. There's a wonderful analogy here. It's as if your camera didn't just take a picture and send the raw pixels to your computer. Instead, the camera itself looked at the image, identified the important features, where the edges are, what's moving, where the brightness changes sharply, and sent a summary of those features rather than the raw data. That's what your retina does. It's not sending a pixel-by-pixel image to your brain. It's sending a processed, compressed, feature-enhanced version. This is why the optic nerve only has about 1 million fibers, despite the retina having over 100 million photoreceptors. The data has already been crunched. And here's a fun detail that most people find a little unsettling when they first hear it. There's a spot on your retina where the optic nerve exits the eye, where all those ganglion cell axons bundle together and punch through the retina on their way to the brain. And at that spot, there are no photoreceptors, none at all, which means there's a spot in your visual field where you are, literally, blind. It's called the blind spot, and each of your eyes has one. You don't normally notice it because your brain fills in the gap, interpolating from the surrounding visual information, essentially making up what it thinks should be there. Your brain is lying to you about the contents of your visual field, and it does it so seamlessly that you never notice. Think about what that means for a second. Your conscious visual experience, the rich, detailed, full-color scene you think you're seeing, is not a direct readout of what's hitting your retina. It's a construction. It's your brain's best model of what's out there, built from incomplete and processed data, filled in with assumptions and predictions. And this isn't just true for the blind spot. It's true for all of vision. The raw data coming into your brain is much sparser, noisier, and more ambiguous than your visual experience suggests. Your brain is doing an enormous amount of interpretive work behind the scenes, and it's doing it so well that you mistake the interpretation for reality. That's a genuinely profound idea, and it's not just a quirk of the visual system. It's a fundamental principle of how the brain works across all the senses. Your brain doesn't passively receive sensory data. It actively predicts what it expects to sense, compares the predictions to the incoming data, and updates the model based on the discrepancies. Some neuroscientists call this the predictive brain, or predictive processing framework, and it's one of the most influential ideas in modern neuroscience. According to this view, most of what you consciously experience isn't sensation at all. It's prediction. Your brain is running a constantly updated simulation of the world, and it only pays attention to the sensory data that doesn't match its predictions, because that's where the new information is. That's where the surprises are. This explains a lot of things you've probably experienced. Why you stop noticing the hum of an air conditioner after a few minutes. Your brain predicted it. The prediction matched reality, so there was nothing new to report. Why you might not notice a friend has gotten a haircut. Your brain predicted their appearance based on memory, and the prediction was close enough that the change didn't flag as significant. Why optical illusions work. They exploit the assumptions your brain uses to construct its model, feeding it data that leads to incorrect predictions. Now let's talk about hearing, because the ear is, if anything, even more mechanically impressive than the eye. Sound is just pressure waves in the air, compressions and rarefactions, regions where air molecules are pushed together and pulled apart, traveling outward from a source like ripples in a pond. And somehow, your ear takes those pressure waves and converts them into the experience of music, of speech, of a door closing in another room, of wind through trees. Here's how it works, and honestly, every step is beautiful. The outer ear, the visible flap of cartilage on the side of your head, collects sound waves and funnels them down the ear canal to the eardrum, a thin membrane stretched tight like the skin of a drum. Sound waves make the eardrum vibrate, and those vibrations are passed along by 3 tiny bones in the middle ear, the malleus, the incus, and the stapes. Those are the hammer, the anvil, and the stirrup, and they're the 3 smallest bones in your body. The stapes, the stirrup, is barely bigger than a grain of rice. These 3 bones form a mechanical linkage that amplifies the pressure of the sound wave by a factor of about 20, which is necessary because the vibrations need to be transferred from air, which is a light medium, to fluid, which is a much denser one. And that's where the cochlea comes in. The cochlea is a spiral-shaped, fluid-filled tube in the inner ear. It looks like a tiny snail shell, and in fact, cochlea is the Latin word for snail. Inside the cochlea, running along its entire length, is a strip of tissue called the basilar membrane. And here's the really clever part. The basilar membrane isn't uniform. It's narrow and stiff at the base of the cochlea, and wider and more flexible at the apex. This means different parts of the membrane resonate at different frequencies. High-pitched sounds cause the base to vibrate. Low-pitched sounds cause the apex to vibrate. The cochlea is essentially performing a Fourier analysis of the incoming sound, decomposing a complex wave into its component frequencies and mapping those frequencies onto different physical locations along the membrane. It's an analog spectrum analyzer, built out of biology. Sitting on top of the basilar membrane are the hair cells, about 15,000 of them in each ear. These are the actual transducers, the cells that convert mechanical vibration into electrical signals. Each hair cell has a bundle of tiny, stiff projections on its top surface, called stereocilia. When the basilar membrane vibrates, the stereocilia are deflected, and this deflection opens ion channels at their tips. Literally, the channels are pulled open by molecular tethers connecting adjacent stereocilia, like tiny trapdoors on springs. Ions flood in, the cell depolarizes, and it releases neurotransmitter onto the nerve fibers at its base. The signal is sent to the brain, and the sensitivity of this system is almost beyond belief. At the threshold of hearing, the eardrum vibrates by about 1/10 of a nanometer, less than the diameter of a hydrogen atom. The hair cells can detect displacements of their stereocilia as small as the width of a single atom. If your ears were any more sensitive, you'd hear the random thermal motion of air molecules. You'd hear the noise of the universe's heat. Evolution has pushed the sensitivity of the human ear to essentially the physical limit. And your brain takes these signals, the pattern of activity across those 15,000 hair cells, telling it which frequencies are present and how loud they are, and constructs from them the rich auditory world you experience. It separates the singer's voice from the guitar, from the drums. It locates sounds in three-dimensional space by comparing the tiny differences in timing and intensity between your two ears. A sound from your right reaches your right ear a fraction of a millisecond before your left, and it's very slightly louder in the right ear, and from those minuscule differences, your brain computes the direction. It extracts speech from background noise. It recognizes a friend's voice in a crowd, all from pressure waves jiggling a membrane the size of your pinky fingernail. And then there's pain. Pain is a different kind of sensory experience, and the nervous system handles it in a uniquely complex way. Pain isn't just a simple signal, "This hurts, make it stop." It's a multidimensional experience that involves sensation, emotion, cognition, and context, all processed by different parts of the nervous system working together. The sensory component starts with nociceptors, specialized free nerve endings found throughout your skin, muscles, joints, and internal organs. Nociceptors respond to potentially damaging stimuli, extreme heat or cold, intense pressure, and certain chemicals released by damaged cells. When a nociceptor is activated, it sends an action potential along sensory nerves to the spinal cord. But here's where it gets interesting. In 1965, two scientists, Ronald Melzack and Patrick Wall, proposed something called the gate control theory of pain. Their idea was that the transmission of pain signals from the spinal cord to the brain isn't automatic. There's a gate in the spinal cord that can be opened wider or closed down by other neural signals. Specifically, signals from non-painful touch receptors, the ones activated by gentle rubbing or pressure, can close the gate and reduce the transmission of pain signals. This is why rubbing a bumped elbow feels good. It's not just distraction. You're actually activating nerve fibers that inhibit the pain signal at the spinal cord level. The gate is being closed, and the brain can close the gate too by sending signals downward to the spinal cord that suppress pain transmission. This is why soldiers in battle sometimes don't feel severe wounds until the fighting is over. The brain, focused on survival, is clamping down on the pain signal. It's why athletes can play through injuries they barely notice until after the game. The pain signal is being generated at the site of injury, but the brain is turning down the volume. This descending pain modulation system uses its own set of neurotransmitters, including endorphins, the body's natural painkillers. Endorphins are chemically similar to opioid drugs like morphine, and they work on the same receptors. When someone says exercise produces a runner's high, that's endorphins binding to opioid receptors in the brain, dampening pain and producing a feeling of well-being. Your body manufactures its own morphine. It's been doing it for hundreds of millions of years. But let's move from how the brain takes in information to how the brain itself is organized. Because the architecture of the brain, the way different functions are distributed across its wrinkled, walnut-shaped surface, is one of the great stories of neuroscience. And like so many great stories, it begins with a tragedy. The year was 1848. The place was a railroad construction site near Cavendish, Vermont, and the man at the center of the story was a 25-year-old railroad foreman named Phineas Gage. Gage was supervising a crew that was blasting rock to clear a path for new railroad tracks. This involved drilling holes in the rock, filling them with gunpowder, laying a fuse, covering the powder with sand, and then using a long iron rod, a tamping iron, to compact everything down. It was dangerous work, but Gage was experienced, and by all accounts, very good at his job. On September 13th, 1848, something went wrong. The powder ignited prematurely, probably because Gage or an assistant forgot to pour the sand, or because a spark was struck between the iron rod and the rock. The explosion drove the tamping iron upward, through Gage's skull. The rod was 3 feet 7 inches long, 1¼ inches in diameter, and weighed over 13 pounds. It entered under his left cheekbone, passed behind his left eye, through the front part of his brain, and exited through the top of his skull, landing 80 feet away. And here's the astonishing part. Gage survived. Not only did he survive, he was conscious and speaking within minutes. He reportedly sat up in the ox cart that carried him to town and spoke to the doctor who examined him. He eventually made a full physical recovery, at least in terms of his motor abilities and his basic cognitive functions. He could walk, talk, remember things, reason through problems. But something had changed. The people who knew Gage before the accident said he was a different person afterward. Before, he'd been capable, responsible, well-liked by his men, shrewd in planning. After, he was reportedly fitful, irreverent, profane, impatient, obstinate, unable to follow through on plans. His physician, John Harlow, famously wrote that the balance between Gage's intellectual faculties and his animal propensities had been destroyed. His friends said he was no longer Gage. Now, the exact details of Gage's personality change have been debated by historians, and some of the more dramatic accounts may have been embellished over time. But the core observation was genuine and enormously important. The tamping iron had destroyed a specific part of Gage's brain, the left frontal lobe, particularly the prefrontal cortex, and the result was a change not in his basic abilities, but in his personality, his judgment, his ability to plan and regulate his behavior. This was one of the first clear pieces of evidence that specific brain regions are responsible for specific mental functions. The prefrontal cortex, it seemed, had something to do with who a person is, with personality, with social behavior, with the ability to weigh consequences and make rational decisions. This idea that different parts of the brain do different things is called localization of function, and it became one of the central quests of neuroscience. And the person who advanced it most dramatically in the 19th century was a French physician named Paul Broca. In 1861, Broca examined a patient named Louis Victor Leborgne, a man who had lost the ability to speak. He could understand language perfectly well. He could read. He was intelligent. But when he tried to speak, the only syllable he could produce was tan. He repeated it over and over. Tan, tan, tan. which is why he became known in the medical literature simply as Tan. When Leborgne died, Broca examined his brain and found a lesion, a region of damage, in the left frontal lobe, in an area now known as Broca's area. Broca presented the case to the Anthropological Society of Paris, arguing that this specific region of the brain was responsible for the production of speech. It was a landmark moment. A specific mental function, speaking, had been localized to a specific spot in the brain. A few years later, in 1874, a German physician named Karl Wernicke identified a different kind of language disorder. His patients could speak fluently. They could produce words and sentences with normal grammar and rhythm, but what they said made no sense. The words came out in the right order, but didn't add up to anything meaningful. And they had difficulty understanding spoken language too. Wernicke found that these patients had damage to a different area, in the left temporal lobe, behind the ear, in a region now called Wernicke's area. So here were two distinct language problems, one involving production, one involving comprehension, caused by damage to two different brain regions. The brain wasn't a uniform mass of thinking jelly. It had specialized zones, and damage to different zones produced different deficits. But perhaps the most vivid, the most striking demonstration of brain localization came in the mid-20th century, from a Canadian neurosurgeon named Wilder Penfield. And his story is worth savoring, because what he did was extraordinary. Penfield was treating patients with severe epilepsy, patients whose seizures couldn't be controlled with medication. The surgical treatment involved removing the specific area of the brain where the seizures originated. But to do this safely, Penfield needed to map the brain's surface, to figure out what each region near the seizure focus did, so he could avoid removing anything critical. And here's the remarkable thing. Brain surgery can be performed while the patient is awake, because the brain itself has no pain receptors. You can touch the brain, probe it, even stimulate it electrically, and the patient feels nothing at the site of stimulation. So Penfield would open the skull under local anesthesia, expose the brain, and then systematically touch different points on the cortex with a small electrical probe while the patient lay there, awake and talking. And what happened was magical. When Penfield stimulated a point on the motor cortex, the strip of brain running roughly from ear to ear across the top of the head, the patient would involuntarily move a specific body part, stimulate one spot, and the right thumb twitches. Move the probe a few millimeters, and now it's the index finger. A little further, and the wrist moves. He could map the entire body onto the motor cortex, point by point, and he found something fascinating about the map. It wasn't proportional to body size. The areas devoted to the hands and fingers were huge, disproportionately large compared to the actual size of the hand. The area for the lips and tongue was also enormous. The trunk and legs got relatively little space. The amount of brain area devoted to a body part corresponded not to the physical size of that part, but to how finely you can control it. Your hands and mouth are capable of incredibly precise, delicate movements. Writing, typing, speaking, playing instruments, and the brain devotes correspondingly more real estate to controlling them. Penfield drew this mapping as a distorted human figure draped over the motor cortex, with enormously oversized hands, lips, and tongue, and tiny legs and trunk. He called it the homunculus, the little man, and there's a matching one on the sensory cortex, which runs parallel to the motor strip. Stimulate points on the sensory cortex, and the patient reports feeling sensations in specific body parts, tingling in the thumb, warmth on the lip, pressure on the foot. And this sensory homunculus is similarly distorted. The fingertips and lips get far more cortical territory than the back or the calf, which is why your fingertips can feel details that your back can't even detect. But Penfield's explorations didn't stop at movement and sensation. When he stimulated certain areas of the temporal lobe, patients reported something far more unexpected. They experienced vivid memories. Or what seemed like memories. A patient might suddenly hear a piece of music they hadn't thought about in years, or relive a specific moment from childhood, or smell a smell from a place they'd once visited. These weren't vague recollections. They were described as vivid, present-tense experiences, as if the patient were actually there again. Penfield interpreted this as evidence that the brain stores detailed, permanent records of experience, complete recordings, essentially, that can be replayed when the right spot is stimulated. Now, we should be careful here, because later research has suggested that Penfield's interpretation was probably too strong. What patients experienced during stimulation might not have been genuine retrieved memories, but rather confabulations, the brain generating a vivid experience on the spot, something that felt like a memory but wasn't necessarily an accurate replay of a real past event. Memory, as we've come to understand it, doesn't work like a video recording. It's reconstructive. Every time you remember something, you're rebuilding it from fragments, and the rebuilding process is influenced by your current state, your expectations, and everything you've experienced since the original event. But Penfield's demonstrations were still immensely valuable, because they showed, in the most direct and dramatic way possible, that specific brain regions are involved in specific aspects of experience. The brain isn't uniform. It's a patchwork of specialized zones, each contributing something different to the total picture of your conscious life. And one of the most striking demonstrations of this specialization came from a series of experiments in the 1960s that are among the most famous in all of neuroscience. The split brain experiments, conducted primarily by Roger Sperry and his student Michael Gazzaniga. To understand these experiments, you need to know one thing about brain anatomy. The brain has two hemispheres, left and right, connected by a thick bundle of about 200 million nerve fibers, called the corpus callosum. This bundle is the main communication highway between the two halves. And in a small number of patients with severe intractable epilepsy, surgeons had discovered that cutting the corpus callosum, severing the connection between the hemispheres, could dramatically reduce seizures by preventing the abnormal electrical activity from spreading from one hemisphere to the other. The patients who had this surgery, the so-called split brain patients, seemed remarkably normal in everyday life. You could talk to them, and they seemed fine. But Sperry and Gazzaniga designed clever experiments to reveal what had actually changed. They used a setup where they could present visual information to only one hemisphere at a time, taking advantage of the fact that each visual field is processed by the opposite hemisphere. Your left visual field goes to your right hemisphere, and vice versa. And here's what they found, and it's genuinely one of the most mind-bending results in the history of brain science. If you showed a picture to the right hemisphere of a split brain patient, by flashing it in their left visual field, the patient couldn't name the object. They'd say they didn't see anything, or they'd guess randomly. But if you asked them to reach behind a screen with their left hand, which is controlled by the right hemisphere, and pick out the matching object by touch, they could do it perfectly. The right hemisphere had seen the picture and knew what it was, but it couldn't produce the word, because the language centers are in the left hemisphere, and the connection between the hemispheres had been cut. The left hemisphere, the talking hemisphere, genuinely didn't know what the right hemisphere had seen. What's even more striking is what happened when the two hemispheres were given conflicting information. In one famous experiment, the word walk was flashed to the right hemisphere. The patient stood up and started walking. When asked why he was walking, a question answered by the left hemisphere which hadn't seen the word, the patient didn't say, "I don't know." Instead, the left hemisphere made up a reason. "I'm going to get a drink," the patient might say, or "I need to stretch my legs." The left hemisphere, lacking the real reason, confabulated a plausible-sounding explanation and delivered it with complete confidence. Gazzaniga called this function of the left hemisphere the interpreter. It's a module that constantly constructs a narrative to explain our behavior, even when it doesn't have access to the real causes. And this has profound implications for how we think about consciousness, free will, and self-knowledge. How much of the time, in a normal, connected brain, is the interpreter doing the same thing, coming up with stories about why we did what we did, stories that feel true but might not be the actual cause? It's a deeply unsettling question, and it's one that the split-brain experiments thrust into the center of neuroscience and philosophy of mind. But perhaps the most important insight to come out of brain mapping in the 20th century, more important in a way than any specific localization finding, was the discovery that the map itself can change. The brain isn't static. Its wiring, its organization, even the amount of cortex devoted to different functions can shift in response to experience. This is neuroplasticity, and it overturned one of the most deeply held assumptions in neuroscience for most of the 20th century. The assumption that the adult brain is fixed, that once development is complete, the wiring is set, and you're stuck with whatever you've got. This assumption was so deeply entrenched that when evidence against it started appearing, many scientists simply refused to believe it. The story of how it was overturned involves several threads, but one of the most important started with a series of experiments by a neuroscientist named Michael Merzenich at the University of California, San Francisco, in the late 1970s and 1980s. Merzenich was studying the somatosensory cortex of monkeys. That's the sensory homunculus we talked about, the strip of cortex that maps touch sensations from the body. He mapped the cortex carefully, identifying which neurons responded to touch on which finger, and then he amputated one finger. Not to be cruel, these were scientifically justified experiments aimed at understanding a fundamental question about brain organization. And what he found afterward was startling. Over the following weeks and months, the cortical territory that had previously responded to the amputated finger didn't just go silent. It was taken over by the neighboring fingers. The brain had remapped itself. The neurons that used to respond to the missing finger now responded to the fingers on either side of it. The map had changed. This was revolutionary, and the reverse was also true. If you trained a monkey to perform a task that required intense use of specific fingers, like pulling a food pellet from a narrow slot, which demands very precise finger movements, the cortical area devoted to those fingers expanded. The more you used a body part, the more brain space it got. The map was dynamic. It was experience-dependent. The brain was rewiring itself in response to how the body was being used. And this isn't just true for monkeys. The same principle operates in human brains. Studies of violin players, for instance, have shown that the cortical representation of the left-hand fingers, the ones doing all that precise fingering on the strings, is significantly larger in violinists than in non-musicians, especially in people who started playing at a young age. London taxi drivers, who spend years memorizing the incredibly complex layout of London streets, a process called the knowledge, have been found to have enlarged hippocampi, the brain structures associated with spatial navigation and memory. Braille readers have expanded cortical representations of their reading finger. The brain is not a machine that's built once and then runs as is for the rest of your life. It's a machine that's constantly rebuilding itself, literally restructuring its connections in response to how you're using it. And this has enormous practical implications. It means that recovery from brain injury is possible, even when significant tissue has been damaged, because undamaged parts of the brain can sometimes take over functions that were lost. It's why physical therapy after a stroke is so important. The intense, repetitive practice isn't just rebuilding muscle. It's remodeling the brain, coaxing neighboring regions to take over for the damaged ones. Neuroplasticity is the basis of rehabilitation. All right, let's shift gears now and talk about a part of your nervous system that you almost never think about consciously, but that is, at this very moment, running some of the most critical operations in your body. This is the autonomic nervous system, the automatic part, the control system that manages everything you don't have to think about. Your heartbeat, your digestion, your blood pressure, your body temperature, the dilation of your pupils, whether you're sweating or shivering. All of it, running quietly in the background, without any input from your conscious mind. The autonomic nervous system is divided into two main branches, and they work in opposition to each other, like the gas pedal and the brake on a car. The first branch is the sympathetic nervous system. This is your fight-or-flight system, the one that gears your body up for action in response to a perceived threat. When something scares you, a loud noise, a near miss in traffic, the shadow of something large moving in your peripheral vision, the sympathetic nervous system kicks in. Your heart rate increases, your blood pressure rises, your pupils dilate to let in more light. Your airways open wider so you can take in more oxygen. Blood flow is redirected away from your digestive system and toward your muscles. Your liver releases stored glucose into the bloodstream for quick energy. Your adrenal glands dump adrenaline, epinephrine, into your blood. Your whole body shifts into a state of high alert, ready to fight or run. This is a magnificent system when it works appropriately, when there really is a threat and you need to respond quickly. But here's the thing. The sympathetic nervous system doesn't distinguish very well between actual physical danger and psychological stress. Your boss, sending a terse email, can trigger the same cascade of physiological responses as a tiger jumping out of the bushes. Your body goes into fight-or-flight mode over things that can't be fought or fled from. And if this happens too often, for too long, the chronic activation of the sympathetic system takes a real toll. Elevated cortisol, increased inflammation, suppressed immune function, high blood pressure, disrupted sleep. Chronic stress is genuinely damaging to health, and the mechanism by which it does its damage runs straight through the sympathetic nervous system. The second branch is the parasympathetic nervous system, and this is the counterbalance. It's sometimes called the rest and digest system, and it does exactly what that name suggests. It slows your heart rate. It promotes digestion. It constricts your pupils. It encourages the body to relax, repair, and conserve energy. After the threat has passed, the parasympathetic system brings everything back down to baseline. Heart rate slows. Breathing deepens. The gut resumes its work. The body shifts from emergency mode to maintenance mode. And the single most important nerve in the parasympathetic system, arguably the single most important nerve in your entire body that you've never heard of, is the vagus nerve. The vagus is extraordinary. Its name comes from the Latin word for wandering, and it's aptly named because it wanders more than any other nerve in the body. It starts in the brainstem, at the very base of your skull, and it runs downward through the neck, past the heart, past the lungs, into the abdomen, reaching the stomach, intestines, liver, and other organs. It's the longest cranial nerve in the body, and it carries signals in both directions, from the brain to the organs, and from the organs back to the brain. Here's something most people don't realize about the vagus nerve. About 80% of its fibers are sensory. They carry information from the organs up to the brain, not the other way around. Your gut, your heart, your lungs are constantly reporting back to your brain through the vagus nerve, telling the brain what's happening in your internal world. This is why you get gut feelings. This isn't just a metaphor. Your gut has an extensive network of neurons, about 500 million of them, and they communicate with the brain primarily through the vagus nerve. When people talk about a gut instinct, or say they felt it in their stomach, there's a real physiological basis for that. The gut is sending signals to the brain, and those signals influence your emotional state and your decision-making in ways that are only beginning to be understood. In fact, the nervous system of the gut is so extensive and so capable of operating independently that it's sometimes called the enteric nervous system, or the second brain. It contains more neurons than the spinal cord. It can coordinate complex patterns of muscle contraction, peristalsis, the rhythmic squeezing that moves food through your digestive tract, entirely on its own, without any input from the brain. If you sever the vagus nerve, the gut keeps working. It slows down, and it loses some of its fine regulation, but it keeps digesting, keeps contracting, keeps doing its job. It is, in a very real sense, a brain in its own right, a brain devoted entirely to the business of processing food. And while we're talking about parts of the nervous system that operate without your conscious knowledge, let's talk about reflexes, because they're a beautiful illustration of how the nervous system can do incredibly useful things without involving the brain at all. The simplest reflexes don't go to the brain. They're handled entirely by the spinal cord. Think about the classic knee-jerk reflex, the one the doctor tests by tapping just below your kneecap with a little rubber hammer. Here's what actually happens. The tap stretches the patellar tendon, which stretches the quadriceps muscle slightly. Inside the muscle, there are stretch receptors called muscle spindles. When the muscle is stretched, the spindle fires, sending an action potential along a sensory neuron into the spinal cord. In the spinal cord, that sensory neuron connects directly to a motor neuron. Just one synapse, one connection. And the motor neuron fires an action potential back down to the quadriceps, causing it to contract. That's the kick. The entire circuit, sensation, processing, response, happens in the spinal cord. By the time the signal reaches your brain and you become consciously aware that the doctor tapped your knee, the kick has already happened. Your brain is informed after the fact. This is a monosynaptic reflex, just one synapse in the circuit. It's the fastest response your nervous system can produce, and it takes about 25 to 50 milliseconds from tap to kick. And it exists for a reason. When you're standing and your weight shifts slightly, stretching the leg muscles, the stretch reflex corrects the imbalance by contracting those muscles immediately, keeping you upright. It's a continuous, automatic stability system, and it works much too fast for conscious thought to be involved. But there are more complex reflexes too. The withdrawal reflex, for instance. Step on a sharp object, and your foot pulls away before you've consciously registered the pain. This reflex involves multiple synapses and coordinates the contraction of some muscles and the relaxation of others. You need to flex the hip and knee to lift the foot, while simultaneously adjusting the other leg to keep your balance. All of this happens in the spinal cord, through circuits called reflex arcs, without any involvement from the brain. It's local processing, fast and efficient, keeping you safe while your brain catches up. And here's something beautiful about how these spinal circuits relate to the rest of the nervous system. The brain doesn't control your movements by directly commanding each individual muscle. That would be impossibly complicated. There are over 600 muscles in your body, and most movements involve the coordinated activation of dozens of them. Instead, the brain sends high-level commands to the spinal cord, essentially saying, "Reach for that cup," or "Take a step forward," and the spinal circuits work out the details. The spinal cord contains pattern generators for common movements like walking, circuits that can produce the alternating, rhythmic pattern of muscle activation needed for locomotion without continuous input from the brain. That's why a cat with its spinal cord severed from its brain can still produce walking movements on a treadmill. The pattern is built into the spinal cord itself. The brain provides the go signal and the steering, but the moment-to-moment execution is handled locally. This is a recurring theme in the nervous system: delegation. The brain doesn't micromanage. It sets goals and delegates the details to lower-level circuits. The spinal cord handles movement patterns. The brainstem handles breathing, heart rate, blood pressure. The cerebellum, that dense, walnut-shaped structure at the back of your head, handles the fine-tuning of movements, the coordination and timing that makes your actions smooth rather than jerky. Each level of the hierarchy handles what it's best suited for, and they communicate with each other through a constant flow of signals, up and down the spinal cord and through the brainstem. And this brings us to something that's worth pausing on: the sheer scale of communication happening in your nervous system right now, at this moment. Your brain contains roughly 86 billion neurons. Each neuron makes somewhere between 1,000 and 10,000 synaptic connections. That means your brain contains somewhere on the order of 100 trillion synapses. Some estimates go higher. To put that in perspective, there are more synapses in your brain than there are stars in the Milky Way galaxy, and at each of those synapses, chemical signals are being released, received, and processed, many of them firing multiple times per second. The sheer volume of computation happening in the 3 pounds of tissue inside your skull is almost impossible to comprehend. And yet, for all this complexity, for all these billions of neurons and trillions of connections, there are basic questions about the brain that we still can't answer. How does conscious experience arise from the activity of neurons? Why does the activity of certain networks feel like something? Why does the firing of neurons in your visual cortex produce the subjective experience of seeing blue, or the warmth of sunlight, or the face of someone you love? This is what philosophers call the hard problem of consciousness. And neuroscience, despite its extraordinary progress, hasn't solved it. We can correlate brain activity with conscious experience. We can identify which brain regions are active during specific experiences. But we can't explain why there is experience at all. Why isn't all that processing happening in the dark, without any subjective awareness, like a complex computer running calculations but nobody home to witness them? Some scientists think we'll solve it eventually, that consciousness will turn out to be an emergent property of sufficiently complex information processing, something that naturally arises when a system reaches a certain level of integration and feedback. Others think there's something deeper going on, something that our current frameworks can't capture. The honest answer is that we don't know, and that's okay. It's a mark of how much we've learned that we can state the question so precisely. But there are things we're learning at an extraordinary pace, thanks to technologies that would have seemed like science fiction just a few decades ago. Let's talk about some of them, because the tools we've developed to study the brain are themselves a fascinating story of engineering and ingenuity. Start with brain imaging. For most of human history, the only way to study the brain's structure was to open the skull and look, which generally required the person to be dead, or at least undergoing surgery. The living, working brain was invisible, locked away behind bone. And then, in the 1970s, A British engineer named Godfrey Hounsfield and an American physicist named Alan Cormack independently developed computed tomography, the CT scan. This used X-rays, taken from many angles around the head, combined with sophisticated mathematics, to construct a cross-sectional image of the brain without opening the skull. For the first time, doctors could see the structure of a living person's brain. They could identify tumors, bleeding, structural abnormalities, all without surgery. Hounsfield and Cormack shared the Nobel Prize in 1979. Then came MRI, magnetic resonance imaging, which uses powerful magnetic fields and radio waves to produce even more detailed images of soft tissue. And in the early 1990s, a technique called functional MRI was developed, which doesn't just show brain structure, it shows brain activity. When a region of the brain becomes more active, it uses more oxygen, which changes the magnetic properties of the blood flowing through it. Functional MRI detects these changes and produces maps showing which parts of the brain are active during different tasks. For the first time, we could watch the brain thinking, in real time, without opening the skull or inserting any probes. And there are other tools. EEG, electroencephalography, uses electrodes placed on the scalp to measure the electrical activity of large populations of neurons. It's been around since the 1920s, when the German psychiatrist Hans Berger first recorded electrical brainwaves in humans, and it's still enormously useful because it has exquisite time resolution. It can track changes in brain activity on a millisecond by millisecond basis, much faster than fMRI. Magnetoencephalography, MEG, measures the tiny magnetic fields produced by neural activity. PET scans track radioactive tracers through the brain to measure blood flow and metabolic activity. And then there are the techniques that don't just observe but intervene, that actually change brain activity. Deep brain stimulation, for example, involves implanting electrodes deep within the brain and delivering precise electrical pulses to specific circuits. It's been remarkably effective for treating Parkinson's disease. Patients who are trembling and rigid can have their symptoms dramatically reduced the moment the stimulator is turned on. It's also being explored for treatment-resistant depression, obsessive-compulsive disorder, and other conditions. The idea that you can modulate specific brain circuits with electrical stimulation and produce profound changes in movement, mood, and behavior tells you something important about the relationship between brain activity and mental life. These experiences, the tremor, the depression, the obsession, are not abstract, free-floating mental states. They are patterns of activity in specific neural circuits, and when you change the circuit activity, you change the experience. There's also transcranial magnetic stimulation, TMS, which uses magnetic pulses delivered from outside the skull to temporarily activate or inhibit specific brain regions. It's non-invasive, it requires no surgery, and it's being used both as a research tool and as a treatment for depression. And optogenetics, a technique developed primarily by Carl Dieseroth at Stanford, goes even further. In optogenetics, specific neurons are genetically modified to produce light-sensitive proteins. Then, by shining light of the right color onto those neurons, typically through a tiny fiber optic cable inserted into the brain, you can turn specific cells on or off with millisecond precision. You can activate a single type of neuron in a specific brain region and watch what happens. It's the most precise tool for manipulating brain activity that has ever been developed, and it's transforming our understanding of how neural circuits produce behavior. And perhaps the most futuristic technology of all is the brain-computer interface, a direct connection between the brain and an external device. The idea is straightforward in concept. Record the electrical signals produced by neurons when a person intends to move, and use those signals to control a computer cursor, a robotic arm, or some other device. The practice is immensely challenging, but real progress has been made. In 2006, a system called BrainGate, developed by a team at Brown University, allowed a paralyzed man to control a computer cursor with his thoughts alone by recording signals from an array of electrodes implanted in his motor cortex. Since then, the technology has advanced dramatically. People with paralysis have used brain-computer interfaces to type messages, control robotic arms with enough precision to pick up a cup of coffee and bring it to their lips, and even regain some degree of independent movement through systems that decode their neural signals and route them around the damaged part of the spinal cord to stimulate their own muscles. This is no longer science fiction. It's engineering. It's neuroscience being translated into technology that restores function to people who've lost it. And it's a reminder of something that runs through this entire story, that understanding how the nervous system works isn't just an abstract intellectual exercise. It saves lives. It reduces suffering. It gives people back abilities they thought were gone forever. And we're still at the beginning. For everything we know about the nervous system, and we know a genuinely enormous amount, vastly more than we knew even 50 years ago, the mysteries that remain are just as vast. We don't fully understand how memories are stored and retrieved. We know that the hippocampus is critical for forming new memories, and that the process involves strengthening synaptic connections through a mechanism called long-term potentiation, and that memories appear to be gradually consolidated from the hippocampus into the cortex over time, and that this consolidation happens preferentially during sleep, which is a lovely detail to mention while you're lying here, since your brain is about to spend the night doing exactly that. Replaying the day's experiences, strengthening the important ones, pruning the rest, quietly reorganizing its own wiring while you're not paying attention. And speaking of sleep, since that's where you're heading tonight, let's spend a little time with the neuroscience of sleep itself, because it's one of those topics where the more you learn, the more astonishing it becomes. For a long time, scientists assumed that sleep was simply rest. The brain was shutting down, conserving energy, doing nothing in particular. That turned out to be spectacularly wrong. Sleep is one of the most active, complex, and purposeful states the brain enters, and we now know that it performs functions that the waking brain simply cannot. Your brain cycles through several stages of sleep throughout the night, in roughly 90-minute cycles. There's light sleep, when you're just drifting off and can be easily woken. There's deep sleep, slow wave sleep, characterized by large, rolling electrical waves called delta waves that wash across the cortex. And there's REM sleep, which stands for rapid eye movement, the stage where most vivid dreaming occurs. During REM, the brain is extraordinarily active, in many ways as active as the waking brain, but the body is essentially paralyzed, prevented from acting out the dreams by circuits in the brainstem that suppress motor commands to the muscles. This paralysis is called atonia, and it's a safety mechanism. Without it, you'd physically act out your dreams, which could be dangerous. In fact, there's a condition called REM sleep behavior disorder, where this paralysis fails, and patients literally punch, kick, and leap from bed in their sleep. Here's what's really remarkable about deep sleep. During slow-wave sleep, your hippocampus, the structure crucial for memory formation, replays the patterns of activity it encoded during the day. The same sequences of neural firing that occurred when you were learning something new are reactivated during sleep, but compressed and sped up. It's as if the hippocampus is broadcasting the day's important experiences back to the cortex, which gradually absorbs them into its own, more permanent storage. This is memory consolidation, and it's one of the most important things sleep does. Studies have demonstrated this beautifully. In one classic experiment, Researchers had rats run through a maze during the day while recording the activity of hippocampal neurons called place cells, neurons that fire when the animal is in a specific location in the maze. Each cell fires in a specific spot, so the pattern of neural activity encodes the rat's path through the maze. Then, when the rat fell asleep, the researchers continued recording, and they found that the hippocampus was replaying the same sequences of place cell activity, the same neural path through the maze, during slow-wave sleep. The sleeping brain was literally practicing the maze, and human studies confirm the same principle. People who study new material and then sleep perform significantly better on tests than people who study the same material and stay awake for the same duration. It's not just that sleep prevents fatigue from degrading performance. Sleep actively strengthens the memories. If you learn a new skill, a piano piece, a sequence of finger movements, and then sleep, your performance the next day is measurably better than it was at the end of the practice session. The improvement happens during sleep, not during practice. Your sleeping brain is rehearsing what you learned, strengthening the relevant synaptic connections, pruning away the irrelevant ones, making the skill more robust and more automatic. There's another remarkable process that happens during sleep that was only discovered in 2012 by a neuroscientist named Maiken Nedergaard at the University of Rochester. She and her team discovered what they called the glymphatic system, a waste clearance pathway in the brain that is dramatically more active during sleep than during wakefulness. During sleep, the spaces between brain cells actually expand. The cells physically shrink slightly, and cerebrospinal fluid rushes through these enlarged channels, washing away metabolic waste products that accumulated during the day. One of the waste products it clears is beta amyloid, a protein that forms the plaques associated with Alzheimer's disease. Sleep is literally cleaning your brain, and disrupted sleep is associated with increased accumulation of beta amyloid and higher risk of neurodegenerative disease. Think about what all of this means. Sleep isn't wasted time. It's maintenance time. It's when your brain consolidates memories, strengthens skills, clears out waste, repairs cellular damage, and reorganizes its own wiring. The circuits that orchestrate this, networks in the hypothalamus, the brainstem, the basal forebrain, have been refined over hundreds of millions of years of evolution. Sleep is so important that every animal with a nervous system does something like it, from fruit flies to whales. No species has ever evolved to do without it. That tells you something about how essential it is, and right now, those circuits are doing their work. The adenosine that's been building up in your brain all day, a molecule produced as a byproduct of neural activity, which accumulates during wakefulness and promotes sleep pressure, is at its peak. Your suprachiasmatic nucleus, a tiny cluster of about 20,000 neurons in the hypothalamus that acts as your body's master clock, has been tracking the light-dark cycle through signals from your retina all day, and it's now promoting the release of melatonin from the pineal gland, that small structure that Descartes once imagined was the seat of the soul. The melatonin is circulating through your bloodstream, telling every cell in your body that it's nighttime, that it's time to shift into the patterns of repair and consolidation and quiet maintenance that define healthy sleep. We don't fully understand how the brain generates emotions, or how it binds all the different sensory streams, sight, sound, touch, smell, taste, into a single, unified experience of being a person in a world. We don't know why anesthesia works. We know which drugs produce unconsciousness, and we're getting better at understanding which brain regions they affect. But the precise mechanism by which they shut down conscious experience while leaving brainstem functions intact is still debated, and we don't understand consciousness itself. How the electrochemical activity of billions of neurons gives rise to the feeling of being someone, to the inner light of awareness, to the experience you're having right now as you listen to these words and feel your body resting and notice the quality of the darkness behind your eyelids. That's the deepest mystery there is, and it's a mystery being pursued right now by some of the most creative minds in science, using tools that would have been unimaginable to Galen or Galvani or Cajal. So let that settle over you. Here's what you carry with you tonight. Inside your skull, right now, 3 pounds of tissue, 100 billion neurons, a quadrillion connections, more complex than anything else we've discovered in the universe, is doing something remarkable. It's running your body. It's holding your memories. It's dreaming up ideas and images and stories. It's taking the raw, chaotic torrent of sensory data pouring in from the world and constructing from it a seamless, coherent experience that you call your life. And tonight, as you drift off, it's going to shift into a different mode, a mode where it replays the day's events, consolidates what it's learned, repairs itself, prepares for tomorrow. Sleep isn't the absence of brain activity. It's a different kind of brain activity, an essential one, carefully orchestrated by circuits in the hypothalamus and brainstem that have been refined by hundreds of millions of years of evolution. And the remarkable thing is that the system doing all of this, the system that manages your heartbeat and your memories, your reflexes and your deepest thoughts, the system that lets you feel the weight of a blanket and contemplate the nature of consciousness, is itself the thing that figured all of this out. The human nervous system is the first structure in the known universe that has studied itself. The brain is the organ that discovered the brain. There's something almost impossibly beautiful about that. An organ made of neurons, firing action potentials across synapses, releasing neurotransmitters, reshaping its own connections, and through that process, coming to understand how neurons fire action potentials across synapses and release neurotransmitters and reshape their connections. We are the universe's nervous system, looking inward, trying to understand itself. And it's been a long journey. From the Egyptians throwing the brain away, to Galen's animal spirits, to Galvani's twitching frogs, to Cajal's exquisite drawings of individual neurons, to Hodgkin and Huxley's squid axon equations, to Lowy's dream about frog hearts, to Penfield's electrical probe and the patient's memories flooding back, to the split-brain patients and the interpreter, to neuroplasticity and brain-computer interfaces, and the tools that are letting us peer into the living brain with increasing clarity and precision. Each generation has peeled back another layer, and each layer has revealed something stranger and more beautiful than the last. And right now, the nervous system that let you follow this entire story, every concept, every name, every analogy, is getting ready to do its quiet nighttime work. The neurons that were active as you listened are about to be replayed. The synaptic connections that encoded this experience are about to be consolidated or pruned, and the whole vast network is about to shift into the slow, rhythmic patterns of sleep. Your cortex will produce the rolling, slow waves that characterize deep sleep. Your hippocampus will replay fragments of the day. Your brainstem will keep your heart beating and your lungs breathing, and for a few periods during the night, your brain will generate the vivid, chaotic activity of REM sleep, and you might dream about what nobody can predict. The dreaming brain is creative in ways that even its waking counterpart can't match. So let your breathing slow. Let the weight of your body sink into whatever's supporting it. The nervous system that has carried you through today, every thought, every sensation, every movement, every emotion, is still at work. It's always at work. And tonight, it's going to take care of you the way it always does, quietly, precisely, beautifully. You don't need to do anything. You don't need to think about anything. The system runs itself. It always has. 3.5 billion years of evolution have made sure of that. Just let it do what it does, and let yourself drift, gently and naturally, into sleep.